Hello everyone, welcome to today's news starter. Let's begin with the prelims practice question of the day. All right, take a look at this question. This question is asked because whatever the contribution of MS Swaminathan for the green revolution of India and for the agriculture of India is extremely important for this year for both prelims and mains. The reason being he died this year, so we need to know about him very well. In the context of the given in the context of Green Revolution of India, the terms Sonalika and Kalyan Sona refers to first option high yielding basmati rice variety, second dwarf rice varieties with saline tolerance, high yielding sugarcane variety, high yielding wheat variety. All the four which were given here have been discovered by Mr. M.S. Swaminathan. These are all his contributions. But what does this name means? These names were given to those varieties of wheat which are high yielding in nature. So option C is correct. Let's go to the first topic of the day, 10,000 Genome Project. For science and tech, almost all the questions are from current affairs both in mains as well as in prelims. So we need to know about the recent inventions, innovations, recent methods adopted by the government, recent achievements in science and tech, etc. 10,000 Genome Project is one of such achievement. What is it? It is an attempt to create a reference database of whole genome sequences out of India. It is a program, it is a scientific program to frame out or to chart out the genome of 10,000 Indians from diverse communities. 10,000 Indian individual and their genomes are going to get charted out with this project. So this is the basic identification. This is the basic idea behind the project. So what exactly is a genome? A genome is the entirety of genetic material of an individual. So if there is a man, the entire different genes present in his body, if you combine together all of them, we call it as the genome of that particular person. So we have different population. India is a very diverse country. Multiple different communities are there, different regions, religions, etc. And people have their own marital relationships. So there is a high scope of multiple different genomic sequences. So we have started a project to identify all these things. So in this particular project, 10,000 Indians from diverse community have been identified and their genomes were charted out. The first time a genome sequencing happened in India in the year 2006. About 20 institutions came together for this, but two institutions were heading it. One, Indian Institute of Science, Bangalore, and Center for Cellular and Molecular Biology, that is in Hyderabad. So these two important organizations were leading the project. This is important. India is a very diverse country because around 1.3 billion population is there and 4,600 different population groups are there. The most important point here is in India, because it is an ancient society, we have been practicing endogamy for a very long time. Endogamy means marrying within the region or within the religion, within a caste community, etc. Because of which certain genes are restricted to certain set of people. So identify it, chart it out, so that we will get to know about the genetics of Indian population in the future if we are to develop certain medicines, or if you are to develop some preventive measures for genetic diseases, it will become easy. So this is the reason we follow genetic and genome programs. What exactly is genome sequencing? How it is done? First thing, before studying about it, we need to know about the differences between DNA and RNA. DNA is deoxyribonucleic acid, RNA is ribonucleic acid. DNA is present within the nucleus of each and every cell, and it will have the code to make or manufacture different proteins with which the body will work. What is the function of RNA? RNA will take the code from DNA to the ribosome in the cell. So in the ribosome, the proteins are manufactured. So DNA is the code, ribosome is the manufacturing place. RNA will take the information from DNA to ribosome. I hope you understand it. Let's take a look at this diagram. It is mentioning about DNA, gene, and chromosome. So this is how a cell is. And in the middle of the cell, we will have a nucleus. Inside the nucleus, chromosome will be there. Chromosome is a double-stranded structure. It will look like this. Chromosome is made of multiple DNA. So DNA will be like this, double-stranded. And it will have all these lines which you see here. These are called as nucleotides. Four different types of nucleotides are there: adenine, cytosine, thymine, guanine. You don't have to know about the details, but at least a basic idea you should know. The strand is made up of sugar phosphate band. So every DNA will carry all these arrangements here are codes. So these code will tell what kind of protein should be manufactured. So a RNA will come and attach with the DNA here, and then it will take the information to the ribosome, and the ribosome will manufacture the protein. So when you talk about genome sequencing, it is finding out the order of DNA nucleotides. So you will take a entire DNA, and then entire DNA will be removed, and then we will identify what are the order of nucleotides. Where is adenine located? Where is cytosine located? All these things will be noted out, and then we will chart it out. So this order will provide information about how the DNA of a person is. Based on that, we will be able to identify what are the genetic problems a person is having, what are the unique genetic properties a person is having. So this is why we do this project. Every human body is made up of 24 different chromosomes. Out of this, 22 chromosomes are called as autosomes, and one pair of chromosome is called as sex determining chromosome. That is either X and Y chromosome, or together X and Y chromosome will also be there. Human Genome Project is the biggest genome sequencing project in the history of mankind. It was started in the United States of America. It was a 13-year long project started in 1990, and they couldn't complete it within 13 years. They took a little extra time. They completed it by 15 years. So who did this? It was coordinated by National Institute of Health and US Department of Energy. So at least basic information you should know. Simlipal Tiger Reserve was in news. Where is Simlipal Tiger Reserve located? It is located in Odisha. Simlipal Tiger Reserve was in news because it is the only tiger reserve in the entire world, not just in India. It is the only tiger reserve in the entire world where pseudo-melanistic tigers are found in wild, not in captivity, in wild. Multiple pseudo-melanistic tigers are found. What is a melanistic tiger? As you know, a tiger will be golden color and it will have black color stripes. A melanistic tiger is when the entire body is converted into black color because of extreme amount of production of melanin. A pseudo-melanistic tiger is when the black stripes become bigger in size so that most of its body is covered with black color. So this is present in Simlipal Tiger Reserve. The reason why the tigers are becoming melanistic tiger is because of inbreeding. Inbreeding means tigers breeding within a small community. Any organism will be able to thrive when they are breeding with different organisms, different set of organisms. For example, you take one tiger from one state and you breed it with another tiger from some other state. There will be high amount of genetic differences due to which it will produce good variety of tigers. But when the tigers are breeding within themselves, there is a high chance that they are breeding within the family. So this will result in recessive genes coming into the prospect. Recessive genes are usually disease-causing genes or not very strong genes. So the offsprings will have certain problems. So melanistic tiger is because of inbreeding. So the Simlipal Tiger Reserve and Odisha government has asked the National Tiger Conservation Authority to ensure some other tigers from some other part of the country should be introduced into Simlipal Tiger Reserve. So there will be more genetic diversity. But for examination point of view, all you need to know is it is in Simlipal Tiger Reserve. We have melanistic tigers in the wild. About Simlipal Tiger Reserve, it is a tiger reserve and a national park in Mayurbhanj district of Odisha. Mayurbhanj district is important. The park is actually a part of Mayurbhanj Elephant Reserve. Mayurbhanj Elephant Reserve is the biggest landmass. In this, Simlipal Tiger Reserve is a small part. And it also includes Hadgad Wildlife Sanctuary, Kuldiha Wildlife Sanctuary. So Mayurbhanj Elephant Reserve is there. Mayurbhanj Elephant Reserve includes the Simlipal Tiger Reserve, Hadgad Wildlife Sanctuary, and Kuldiha Wildlife Sanctuary. These points you should be knowing. Pseudomelanistic tigers are only found in wild in Simlipal Tiger Reserve in Odisha. According to the government data, more than 60% of Simlipal tigers are black. Result of inbreeding in the present tiger population, that is the reason. So Odisha government has written to National Tiger Conservation Authority, NTCA, which is the authority to take care of the tiger of the country, to consider introducing female tigers from other landscapes so that breeding will happen with different genetic diverse population. Obelisks. Obelisks are very important. It is a new discovery, so you need to study about it. Before studying about obelisk, we need to know about two important things. One is viruses or virion and viroids. Viruses are virion. Generally, what is the genetic setup of virus? A virus will be having a DNA or RNA. So either it will be a DNA-based virus or RNA-based virus. Surrounding this DNA or RNA, we will have a protein coat. In some cases, there will be a lipid. Lipid means fat coat. So DNA will be there, RNA will be, or RNA will be there. Surrounding it, there will be protein or fat. They will get into the body of a host, a human being or animal or plants or anything. They will get into the body, and then they will get into the cells of that animal, and then it will release its own DNA inside the cell. So this DNA will survive on those cells, it will reproduce, and then it will attack all the other cells. So this is how generally virus work. There is another species called viroids. Viroids are a little different from viruses. Viroids have naked RNA only, just RNA. They do not have proteins around it. They do not have lipids around it. So it is very difficult to understand how they survive. Because when you have protein or lipid around the RNA or DNA, it gives you energy. So it can survive. But in case of a viroid, it just have a RNA or DNA. There will be nothing around it. So it is completely inactive. It will become active only when it enters the body of a host animal. So it is very different from a living being. We can't even call it as a proper living being. We can't call it as a non-living being because it is inactive. It looks like a completely dead material, but once it enters a body, it will become active. So viroids are like this. RNA would enter a cell as RNA, force the cell to make copy of itself. So this RNA will get into the cell and then it will tell the nucleus to make the copy of this kind of RNA, and it will make. The new RNA would infect the other cells, and based on it, it will start proliferation. But now the scientists have identified another different organism. It is called as obelisks. Obelisks are identified in human gut and in human mouth also. So what does this obelisk do? They are present in human gut and mouth. They are rod shaped and they also have RNAs, but they are not similar to viroids. What is the difference? The RNA was longer than the RNA of viroids, and they appear to code for two proteins. So this point is more important. In case of a viroid, it doesn't have any code, just the RNA exists. But in case of obelisk, they have a code also inside it. So this is the basic difference. About it, there is no complete study. It is still under research. So we will get more information about it later. But for now, at least you need to know what obelisk means. Mains question of the day, what is genome sequencing? Today in this session, we have explained in a detailed manner what is genome sequencing. You can write about it. What steps have been taken by India in genome sequencing? We have explained about the 10,000 genome project also. You can write about it. Prelims point of view, Shanti Niketan is very important because recently there was an issue that a lot of trees around Shanti Niketan were cut by the government, and they have raised some complaints about it. So what exactly is Shanti Niketan? Shanti Niketan was a town located in Birbhum district of West Bengal. It is recently included in UNESCO World Heritage Site. Shanti Niketan was built up completely by the Tagore family. It was identified by Debendranath Tagore. Bu he went to a place called as Bhubadanga. Bhubadanga is located in West Bengal. So he went to a place called Bhubadanga, and then he renamed it as Shantiniketan. He built a house there, and later he understood that this place is very beautiful. So Rabindranath Tagore, who is the son of Debendranath Tagore, he established a school there. And he established a school based on the ancient Indian Brahmacharya model, where students can come, they can stay there, they will understand the lifestyle, they will understand the livelihood, they will understand the culture, and they will study the subjects also based on Western as well as Indian model. It was later established as Vishwa Bharati University. It was given UNESCO World Heritage Site status because it teaches not just education, it talks about human value, architecture, arts, town planning, landscape, etc. So all these values together combined, we consider it as a very important UNESCO World Heritage Site. So from prelims point of view, you need to remember the details. That will be all for today. All the best. Study well. Let's meet tomorrow.